Welcome to this ProImmune podcast. My name is Margot, and in this presentation I will introduce you to our ProT2 MHC Class 2 Tetramers, which are powerful tools for detecting antigen-specific CD4 T cell responses. Over the past couple of decades, MHC tetramers have emerged as the technology of choice for monitoring epitope-specific CD4 T cell responses. So what is a ProT2 tetramer? A tetramer consists of 4 peptide-loaded MHC class 2 molecules tetramerized with fluorochrome-labeled streptavidin. The engagement of CD4 T cells via their T cell receptor with the MHC peptide complex enables their visualization, enumeration, phenotypic characterization, and isolation from ex vivo samples. There are many applications to the ProT2 tetramers. They are mainly used to identify and enumerate epitope-specific CD4 T cell populations by immunohistochemistry and flow cytometry. For a detailed characterization of the observed antigen-specific T cell population, our ProT2 tetramers can be included in a protocol using other labeled antibodies, allowing the immunophenotyping of epitope-specific memory subpopulations, for example. Tetramers can also be combined with intracellular staining of cytokines, for example, thus enabling the identification of T cell helper subset the epitope-specific population belongs to. In addition, ProT2 tetramers are useful tools to isolate epitope-specific populations using a cell sorter that can purify the fluorochrome-labeled cells within the appropriate gates. Another interesting application of tetramers is their use to activate epitope-specific T cells ex vivo and clonally expand them. Because they are composed of the MHC molecule and the peptide, which are both essential for antigen recognition and subsequent T cell activation, tetramers can be used in conjunction. Tetramer staining protocol is very simple, with just a few steps required to stain your cell sample. After incubation of your cells with titrated tetramer and a wash step, optimally titrated antibodies like anti-CD4 are added for another 20 minutes. After a further 2 washes, your cells are ready to fix and analyze by flow cytometry. The analysis is performed on the live lymphocyte population using the forward and side scatters to exclude dead cells and debris. A density plot of tetramer versus CD4 is then drawn as shown here. The antigen-specific population of interest, which is tetramer positive, CD4 positive, is shown in the upper right. This is an example of tetramer staining performed on CD4 positive T cell population cultured for 13 days in the presence or absence of influenza peptides. In the peptide-stimulated samples, you can see a clear, isolated positive population in the top right quadrant, demonstrating that our tetramers have successfully detected the antigen-specific T cell population. The bottom dot plots are negative controls using HLA mismatched donors to confirm specificity of staining and low background. This data from Professor Georg Lauer's group at MGH Harvard University displays the evaluation of ex vivo HCV-specific CD4 positive T cell responses using tetramers loaded with both DR1 and DR4 restricted peptides. Epitope-specific CD4 populations are detected at frequencies as low as 0.054%, demonstrating the high sensitivity of this technology, which detects populations at very low frequencies without it being unspecific staining. Finally, this is an example of clonal T cell expansion detected by our tetramers. This data was kindly provided by Professor Ellie Barnes at the University of Oxford. Cells here were stimulated with a peptide pool, and their specificity was characterized using an HCV-specific tetramer. As you can see, a large HCV-specific population was detected after 12 days of culture and expanded after 22 days, increasing from 22.9% to 29.8% of CD4 population. As expected, no staining is observed in the HLA mismatched donor in the bottom plot. Our ProT2 Class 2 tetramers are ready to use. They can be labeled with RPE or APC fluorochrome and are provided in vials of 50 or 150 tests. Over 50 catalog items are available off the shelf, bearing the most common epitopes of various diseases such as EBV, CMV, and cancer. Negative controls loaded with a clip peptide are also in stock. Alternatively, if you are seeking a combination of peptide HLA molecules, which is not available in our catalog items, we can also custom make the tetramers for you using a broad range of HLA alleles. All our tetramers are guaranteed for 6 months when stored according to guidance, allowing you to use the same batch over a long period of time. Catalog items can be dispatched within 3 weeks and custom tetramers in 7 weeks. In conclusion, why use ProT2 Tetramers? They are the most widely used commercial MHC multimer for detection of CD4 positive specific responses and can be used in various assays and experiments. They have an excellent design and confer uniform, specific bright staining while giving you a choice between RPE and APC labeling. Importantly, we pride ourselves with our stringent quality control checks to ensure the excellent quality of our products delivered to you. We offer a wide range of stock items, but are also expert in the synthesis of custom tetramers. Finally, our ProImmune team can offer expert technical support and advice, so do not hesitate to contact us. Please visit our website for more details about our ProT2 Tetramers and other technologies we specialize in. Alternatively, feel free to get in touch with us by email or by telephone for any inquiry. We will be happy to help. Thank you for your attention.